What is gene therapy? Human gene therapy seeks to modify or manipulate the expression of a gene, or to alter the biological properties of living cells for therapeutic use. Gene therapy is a technique that modifies a person's genes to treat or cure disease. Gene therapies can work by several mechanisms: replacing a disease-causing gene with a healthy copy of the gene, inactivating a disease-causing gene that is not functioning properly, introducing a new or modified gene into the body to help treat a disease. Gene therapy products are being studied to treat diseases, including cancer, genetic diseases, and infectious diseases. There are a variety of types of gene therapy products, including plasmid DNA. Circular DNA molecules can be genetically engineered to carry therapeutic genes into human cells. Viral vectors. Viruses have a natural ability to deliver genetic material into cells, and therefore, some gene therapy products are derived from viruses. Once viruses have been modified to remove their ability to cause infectious disease, these modified viruses can be used as vectors, vehicles, to carry therapeutic genes into human cells. Bacterial vectors. Bacteria can be modified to prevent them from causing infectious disease, and then used as vectors, vehicles, to carry the therapeutic genes into human tissues. Human gene editing technology. The goal of gene editing is to disturb harmful genes or to repair mutated genes. Patient-derived cellular gene therapy products. Cells are removed from the patient, genetically modified, using a viral vector, and then returned to the patient. Here we have this lovely image. In vivo. Lipid nanoparticles, which we will learn more about in the next episode. Direct delivery to patient using viral or non-viral delivery vehicle. And on this side, we have ex vivo. Extract the stem cells. We have DNA, RNA, proteins. Then we augment the cells and introduce modified cells back into the patient. We are on the official FDA government website. Let's go over here and let's see what Pfizer has to say. What's next for mRNA? The global pandemic showed the potential of mRNA technology to transform medicine in the coming years. The versatility of messenger ribonucleic acid, or mRNA, can be overemphasized. The same technology that has helped protect people around the globe from COVID-19 may lead to the potential treatments for rare disease and help prevent other infectious diseases. While Pfizer is currently focused on developing mRNA vaccines to help prevent the flu and shingles and to treat rare diseases, it continues to explore other opportunities where technology could make a transformational impact on patients and where its expertise has the potential to accelerate the pace of innovation. If we look forward and really let our imaginations go and start marrying unmet medical needs to this new toolkit that we've developed over the past couple of years, it's going to be really exciting. Prevention. Well before the global pandemic, when mRNA was a lesser-known technology, Pfizer and BioNTech had already been collaborating to make a better flu shot. With traditional vaccines, scientists must work months in advance to predict which influenza strains will be dominant in the upcoming season. And depending on how well the flu shot matches the season's prevalent strains, its effectiveness can vary year to year. But because mRNA technology can be used to produce vaccines more quickly than conventional vaccine manufacturing technologies, Scientists expect it can help create a shot that better matches the season's flu strains and, as a result, may offer better protection. Another key benefit of mRNA technology is its flexibility, which may allow scientists to quickly edit vaccines. For viruses like the flu that are constantly changing and requiring vaccines that need to be updated each year, once a path is established with the FDA or other regulatory authorities, scientists may be able to swap out the mRNA instructions to match with the then circulating variants. In addition to the flu shot, Pfizer and BioNTech are collaborating on a project aimed at producing the first mRNA vaccine for shingles, a painful infection caused by the same virus as chickenpox. Nearly 1 in 3 adults will experience shingles at least once in their lifetime, with episodes lasting 3 to 5 weeks. The new shingles vaccine development program will leverage learnings from the company's COVID-19 vaccine in addition to Pfizer's research on anti-agents, substances that trigger an immune response. Building upon their COVID-19 vaccine partnership, Pfizer and BioNTech are collaborating to develop the first mRNA shingle vaccine. But for the mRNA flu vaccine, more things need to be considered, says Pirada, Vice President of Viral Vaccines at Pfizer. For one, the current flu vaccines are designed to protect against 4 different flu viruses, so any mRNA vaccine will likely need to also provide protection against those 4 viruses. We're really trying to find the best design that gives you the best immune response, and that's something that needs to be tested empirically. Therapies. Looking beyond vaccines, mRNA technology may also help patients with rare diseases. In collaboration with biotech company Beam Therapeutics, Pfizer scientists are developing mRNA technology as a new approach to gene editing, a form of gene therapy. Gene editing works by delivering quote-unquote tools encased in lipid nanoparticles that have the potential to add, remove, or correct faulty genes in patients with rare diseases, providing a potential one-time treatment for these debilitating conditions. With this next-generation technology, mRNA encodes instructions on how to make the tools that could repair single gene mutations. These instructions are accompanied by a guide, RNA, that directs the tools to the precise location that needs editing. This is Precision AI Healthcare. mRNA technology is a good fit for gene editing. We want to make these editing proteins for just a short period of time to modify the genome, and producing the editing enzymes transiently helps to reduce the potential for off-target effects. We're going to go over here, and we're going to read Oversight of Human Genome Editing and Overreaching Principles of Governance. Oversight of human genome editing fits within the overreaching framework of oversight of gene therapy. That framework is embedded within the larger context of international conventions and norms for protection of human rights, and more specifically, for research involving human subjects and clinical care. From these international instruments, one can derive principles of governance of genome editing that have general application within the United States and across the globe, and are reflected in the specific statutory and regulatory rules that are adopted by various nations.
They say they are here to save us and love us, but for some reason, all our rights went out the window when they decided to go ahead without our permission. Vaccines, it's a biocyber interface for intra-body communication. They updated with nano networks routing through the body. mRNA is not a vaccine, It's a bio-nano machine.